Hello everyone, this is Joe, a Mingdao high school student doing synthetic biology. You love food. Well, in fact, everyone loves food, such as hamburgers, sandwiches, and potato chips. We produce 11 billion metric tons of waste annually, just by eating. Pork, fish, wine, vegetables, juice, milk, you name it. Food waste can be converted into fertilizers. However, many spoiled bacteria grow on them and leave strong odors. And why, you ask? Vegetables grow arena and pseudomonas, producing hydrolyzed polymer from pectin and causing slime. Fish cultures Pseudomonas putrefaciens, transforming amino acids, for example, cysteine and methionine, into hydrogen sulfide and producing sulfidy off odor. Milk cultures Pseudomonas fragi, transforming phospholipid to esters and making fruity off odor. Meat feeds Enterobacteria ac and lactic acid bacteria, digesting amino acids into ammonia and produce odors of ammonia, obviously, and putridness. It also feeds Brochostrix stomosphecta with glucose, making acetoin and diacetyl and producing a cheesy off odor. The Cycloblasis acidoterrestrus likes sugars in juice. It produces 2-mesoxyphenol and a medicine off odor. Not that we drink wine, but wine grows Penicillium brevicompactum, digesting 2,4,6-trichlorophenol to trichloroazinol and making musty odor. These are the reasons why food waste smells bad. We decided that the smell has to go, so we took out our biobricks and made some designs. Design 1. Attack. Antimicrobial peptides with net positive charge can attack an ionic membrane of bacteria to form channels or spores, resulting in a leakage of cell contents and cell death. One producing AMPs has NPRF to modify L-lysing and the membrane charge. Sacropin from moths and manganin from frogs were synthesized onto the biobrick, along with OMPA secretion signal to direct them to the medium. Design 2. Decomposing. Stenotrophomonas maltophilia is a soil bacterium and has been proved to be beneficial for plant growth and health. Extracellular decomposing enzymes were cloned from them. Design 3. Self-destruction. CCBD is a bacterial toxin targeting DNA gyrase and inhibiting DNA replication. The gene expression is regulated under lac operon and repressed by lac-I. Now we can proceed to gene cloning. First of all, we run the PCR to amplify the genes. We took a DNA template and some primers, denatured them at 95 degrees Celsius, and annealed the primers to the template. The DNA polymerase then extends the template and amplifies the gene fragments at 72 degrees Celsius. Finally, the PCR product was stored at 4 degrees Celsius. Next, the samples were left on a rack, and we were ready to run a DNA electrophoresis test. Put agarose gel into the tank and mix floating dye with samples, followed by loading them into the well. After 30 minutes, we transferred the gel into the B box and turned on the blue light. Voila! The band showed itself. We let the amplified DNA fragments and the vector digest using restriction enzymes and ligated them with DNA ligase afterwards. Now, what we get here is a recombinant DNA. Next, transformation was ready to go. We put the competent cells in ice and prepared a 40 degrees water bath. The competent cells and ligation products were incubated for 1 minute in water bath and was sent back into the ice. We have created a super transformed E. coli with biobricks. The super bacteria were left to culture at 37 degrees overnight and were transferred to food waste samples. The spoilage bacteria would be eliminated by antimicrobial peptides produced by our super bacteria. Next, the super bacteria would produce decomposing enzymes to digest food wastes. And finally, a suicide bomb in a super bacteria would be induced to prevent impact to the natural environment. And now we have finally created a odorless fertilizer that can be used to fertilize plants and anything that needs fertilizing, well, obviously, right?